Welcome back, everybody, to round 2 of our summer lecture series. It has been duly noted that we will order more pizza for the next one. I don't think we've ever had the room that's full before for these, so this is really great that we weren't eating out. So today, Erin's going to talk to us a little bit about how to image beyond those traditional 3 or 4 colors that they're often stuck with on a light microscope. And Erin was so eager to do this presentation that she spent the last week working on it in Paris and flew in yesterday just to be here today. So we really appreciate all that hard work.
Yeah. There was one GIF that's been, or actually video added to the presentation that I found while in Paris. So it is marginally true that I was working on it in Paris. So yeah, today I want to talk about fluorescence microscopy, but in the context of trying to image multiple colors and the kinds of considerations that you want to think about when you're picking fluorophores. It's becoming very common for people to image many, many colors in a given piece of tissue. And we get a lot of questions about which ones to choose, how to combine them. And so I can't answer the question individually for everyone, but hopefully give you the tools to go find out for yourself what the best combinations are. So first, to frame the motivation for using fluorescence and using many colors in fluorescence is first to show you this image, which is a transmitted light image of some cells on a slide. And you can see the cells. You might even be able to tell some subcellular compartments from others, so maybe you can tell where the nucleus is in some of these cells. But you might have a hard time, even with your own eyes and manual segmentation, segmenting these cells from the background, and a computer would have an even harder time. Speaking from strictly threshold-based segmentation, we won't get into deep learning-based segmentation. Those might have an okay time segmenting this image or finding the nucleus. But based on your eyes or computer's threshold alone, you would have a hard time segmenting this image. And so that's one reason why fluorescence is so powerful, is because it introduces more contrast into our image. So we have a black background, and here there are 3 different things labeled, as are hard to tell with the projector, but 3 different things labeled in this image that tell you something, not just about, it doesn't just introduce contrast in the image, but it also tells you specifically where something is located. So you've labeled the nucleus with something, or you've labeled actin with something, and that something is a fluorescent molecule. So you can tell it apart from the rest of the stuff. And this is, of course, incredibly powerful. It's why fluorescence has been so popular in biology, because we're not able to just see a cell, but we're able to see where a protein is localized in the cell, or even an entire tissue, an entire organism. And the ability to image, say, 3 colors, like in this image, or even more than that, has really been aided by the explosion of different dyes and fluorescent proteins and the development in this area to allow us to pick and choose any combination of fluorescent molecules that we so desire. And it's been matched by, in the model organism field, having many, many proteins already genetically engineered to be expressed in a particular cell type or tagged for different proteins. So you have mouse lines and fly lines and fish lines that you have at your disposal to use these fluorescent proteins. So in some cases, the fluorescent protein is chosen for you. In other cases, you have a huge range of flexibility to use whatever dye of interest you have to do, say, your immunostain or your RNA-scope assay. And so with so many options, we get a lot of questions on how to combine these different fluorophores of interest. And so that's the idea of today's talk, is to sort of give you some ideas of how to combine these in an appropriate way, and some sort of computational quality control methods to check your work and see that an experiment is going well or is not going well. So to start, let's review fluorescence. So here I'm showing you a simplified Jablonski diagram where we have some electron that's at a ground state that can then be excited with a photon that has a particular energy or has a particular wavelength. And that electron will be excited up to a new state here. It will relax by releasing some vibrational energy, and then in some cases a photon can be emitted, and that energy will be released, and that electron will fall back down to the ground state. And because of this loss of vibrational energy, the wavelength or the energy of this light is different than the energy of this released photon, and so it has a different wavelength or a different color. And this allows us to separate our excitation light from our emission light. And so in that simplified diagram, you saw a single color of excitation and a single color of emission, but you're probably familiar with looking at excitation and emission spectra. And what that means is that it's not just a single wavelength that excites your fluorophore of interest or is emitted by your fluorophore of interest, but it's a spectrum of different wavelengths. And one important thing to remember is that if you excite at various points in your excitation spectrum, it's not that you can directly predict exactly what the wavelength that will be emitted is. You still get the full emission spectrum, or we have the possibility of getting the full emission spectrum, but the height of it is dampened whether you've excited at the peak of your excitation spectrum or somewhere on these sort of lower, less efficient ends of your excitation spectrum. And so this is the basis of fluorescence and our ability to separate, for example, our excitation light from our emission light. And if you want a review of how this works component-wise in a fluorescence microscope, we have a previous Lunch and Learn lecture that Doug did that's on our YouTube channel that goes over sort of the specifics of the light path of a fluorescent microscope and how we actually functionally do that. But of course, I'll give some examples in this lecture as well. And I was going to say, well, I'm not going to go over the sort of development of these different dyes. I showed you that plot of different Alexa Fluor dyes, but while I was on vacation perusing Microscopy Twitter, I found this great video from John Grimm. Let's see if this will exit. And John is a scientist at Jamelia. And he's involved in developing some of the Jamelia Fluor dyes that I think some of you have been using. And he made this great video to show you, he's a chemist, so better at explaining the development of these dyes than I am. And I just wanted to go over it because it shows you how you can make small structural changes in a molecule and change the excitation wavelength of that light. And the first example is actually taking you through just what excitation and emission is. So you have some absorption of a photon, some of that energy is lost to vibration, and then the photon is re-emitted, but it's a slightly different wavelength. This symbol is for, we'll go over it in a second, but think of it as sort of the efficiency with which you release that photon. So he made a small structural change and improved the yield of that molecule. And so making a small structural change shifts the excitation wavelength slightly higher. And he's going to sort of iteratively make structural changes to shift that excitation curve to the right, to the red end of the spectrum. And so this is how people who are doing dye development, more experienced in chemistry than I am, are able to take you from a single molecule that is fluorescent and wiggle it back and forth in the visible spectrum and end up with this entire library of Jamelia dyes or Alexa Fluor dyes or atom dyes. And so some of the diversity that we've seen in the field is because of this kind of chemistry. So I just wanted to sort of appreciate the work that So when you consider what dyes or proteins to use, even if you're not combining them with one another, it's still interesting to think about some of the differences that individual dyes or proteins might have based on the different wavelengths that they are excited by or the different wavelengths that they emit. And the first reason for this is because when we consider our resolution, this smallest resolvable difference, we calculate this including a term for wavelength. And so using lower wavelengths of light will lower the resolution, will improve the resolution, but will lower this smallest resolvable distance here. And so even if you're choosing a single fluorescent protein or dye, it might be interesting to consider the resolution that you need to achieve and what wavelength you're using for that particular label or probe. And this NA term, hopefully many people in the audience are familiar, is your numerical aperture, which describes the half angle or is calculated using the half angle, sine of the half angle of this cone of light that's collected by your objective. The higher your numerical aperture, the higher your resolution. So the smaller this smallest resolvable distance gets. But the other term that I'd like you to pay attention to is this n here. And this n describes the refractive index of the medium that you're imaging through. And the refractive index compares the speed of light traveling through that medium versus the speed of light in a vacuum. And so this number is pretty larger than 1 in the media that we're describing because light is slowed down traveling through some medium that's not a vacuum. And this seems to not have a wavelength term in it, but in fact the refractive index is different for different wavelengths of light. And so you can describe a process called dispersion, or the dispersion that happens in a medium like a type of glass, by comparing the refractive indices for yellow light, blue light, red light, etc. And the different dispersion, the different refraction of that light within a medium is important when we consider where our light is focused. So if we look at the lens maker's formula, which describes the focal length of a lens, it considers not only the radii curvature of the lens, but also the refractive index. And if this refractive index is changing for different wavelengths of light, then light will be focused at different points relative to that lens. And for the most part, we have objective lenses that correct for this. So we can actually use the different dispersion present in different materials to make combinations that allow for an effective lack of dispersion so that all colors are focused to the same point. But there are still chromatic aberrations, and so this becomes still a problem if you're trying to perform something like super resolution microscopy, where you're going under the diffraction limit to localize points in your sample. And so this does become relevant when you're combining multiple colors. And then finally, still relevant even if you're just using a single fluorescent dye or protein, is the scattering of that light through your tissue. So again, we have lectures on tissue clearing from Doug on our YouTube channel, but we also have this great review paper that he wrote with our faculty director, Jeff Lichtman, and this goes into great detail on how to mitigate the scattering of light within a sample by using tissue clearing. But it's important to consider that the scattering of light is wavelength dependent. And so in this case, it's dependent on wavelength to the power of -4. So changing the wavelength that you're using dramatically changes the amount of scatter that you have in your tissue. So this is why we frequently use photon excitation, a long wavelength of light, to penetrate deeply into tissue. And so this wavelength dependence is an important consideration when you're picking a dye of interest to image deep into the brain, for example. Okay, so most of those were relevant to even picking individual fluorescent proteins or dyes, but we want to think about the combination of many dyes, let's say. And remember that we talked about separating the excitation and emission light, but also we want to separate the many excitation lights and the many emission light paths that we have from the many dyes that we're using. And one way of doing this is using a filter cube. And so in this case, you have an excitation filter that filters your excitation light to be the wavelengths or band of wavelengths that you want. And you have an emission filter that separates this, that only selects a band of wavelengths that hopefully covers perfectly your emission spectrum. And there's also a dichroic mirror that allows your excitation light to be reflected down to your sample and that emission light to pass through that mirror and get to your detector. But I'm going to focus for the most part on your excitation light and your emission light, so your excitation and emission filters today. And so this is the sort of basis that I want you to have in your mind as we go through some examples of how to consider the separation of two nearby fluorescent dyes or proteins. Okay, so to start, we're going to have some, two nameless excitation and emission spectra. So we could say this is something like 488 and 568. I think those are the ones that I pulled. And you can see that they're, so I'm showing you in green the excitation and emission for 488, let's say, and we'll just call it green, and the excitation and emission for say, red in these reddish colors. And on the left, you would have your excitation. On the right, you would have your emission, because remember, your emission light is always longer wavelength, lower energy than your excitation. And the goal of this experiment is we want to image some neurons, let's say, and the processes are labeled in our green color. The cell bodies are labeled in our red color. And so ideally, we get an image that looks perfect. We only have red in our cell bodies, and we only have green in our processes. And so to simplify this just a little bit, let's move our excitation light over. We'll remember that it's there, but we want to focus on our emission spectra here. So what we want to do is we want to excite our green molecule first. We'll start with that. And we want to collect the emission from our green fluorescent dye. And ideally, we get this image here. So remember that this overlap that we see here, that might seem like, it's not that big of a deal, but how much should I worry about that little tail? Might be a problem. But remember, we're exciting here. And so if you remember that diagram from the beginning on where you placed your excitation within that excitation spectrum, that changed the height of this curve. Right now, these are just normalized to 1. They're all at the same height. But in fact, this should be down here because we're not exciting it very efficiently. And so when you want to compare the overlap of different emission spectra, which is something that people do all the time, they bring in 2 dyes and they're worried that there's some part of the tail that's overlapping, it's important to consider how efficiently are you exciting either of them relative to one another, because the heights of those curves are not actually always at 1. Okay, so now this tail seems like even less of a problem. We don't want to worry about it at all. Okay, but what if this is our sample, and what if it looks a little bit more like this, where we've labeled some green things and some red things, and let's say we have way more of our red probe. So we put 10,000 times the concentration of our red secondary compared to our green secondary. So pretend I could put that many little Y's in my illustrator drawing. Then what you would have is not a scenario where the red curve was significantly lower in magnitude than the green curve, but instead you have something like this, that even though you're exciting the green dye most efficiently, you have way, way more of your red dye, and so that little bit of overlap starts to become a bigger deal. And this is something that we see sometimes in samples where you've picked dyes appropriately, they look well separated, and yet the preparation of the sample is done such that you have way more of one probe than another. And that can be a lasting problem even if you've picked 2 dyes that seem relatively separable. And so then you get an image that looks a bit more like this, where the red is starting to bleed through into the green, and it looks like no problem. And it's not just the amount of probe that you put in your sample. There are also fundamental differences in the dyes that you've chosen. So we sort of talked about the differences in resolution and scattering based on the wavelengths of light, but the individual molecules themselves have properties of how efficiently they absorb or emit light. And so for a given dye, we can describe the quantum yield or the extinction coefficient of that dye. So the efficiency with which light is absorbed, so this first step here. So if you put a photon near that molecule, is it going to absorb that photon? Is it going to absorb that energy? And the quantum yield is the fraction of the time that this emission light is actually emitted as a photon, as the fluorescence that you want, versus some non-radiative decay. So imagine 2 molecules bumping into each other, and that energy is lost by some other mechanism than by an emitted photon. And so ideally, this is 1, that anytime a photon is absorbed, it's emitted as fluorescence, but these numbers are typically slightly under 1 or well under 1. And so the brightness of your dye is proportional to the product of these 2 values. So how efficiently are you absorbing a photon and how efficiently are you emitting a photon? And so let's imagine the same scenario where you maybe didn't have 10,000 times as much of your red probe, but maybe the red dye that you chose was 10,000 times better than the green dye that you chose, for example. And so this can affect your brightness and thereby change the heights of those curves. Okay, so let's imagine this same scenario, our sort of ideal scenario, where these are pretty well separated dyes and we have equal numbers of both of them. And this is the image that we get. And you can't really tell on this projector, but I've put a little bit of bleed through just to represent this little tail of the curve. So you would say probably this tail doesn't matter. There's not a lot of bleed through, but we'll say there's some small amount. And so if we look at the display curve that we talked about actually in the last lecture, where let's pretend this is an 8-bit image, and any pixel can have a gray value up until 255. That could be a different number, a higher number if you had a higher bit depth, but this will pretend it's an 8-bit image. And what this line is representing is if we have some lookup table of the values, how intense that green color is, then this top 255 value would be the brightest green pixel, and if we had a value back here, it would look more like a black pixel, okay? And so right now we're seeing the full dynamic range, and this was made in Illustrator, so these are all the same gray value, but we're going to pretend that we see a nice histogram of gray values. And we have a small amount of bleed through, so there are a few pixels that have these very, very low gray values. So that's representing our bleed through. So we see it in the green channel, but I'm going to color it red because it's the red bleeding through into the green. Okay, so if we saw this image, like this would be fine. We can clearly see where the processes are. And they're also spatially separated from the cell body, so this is probably a pretty good dye selection. But if I changed my display curve so that now everything above here was that brightest green pixel, and my only dynamic range that I'm really seeing as far as my lookup table matched to my the real gray values of my image, I'm only seeing these differences here. And so my image would look a bit more like this. So sort of hard to represent saturation in Illustrator, but these would look totally saturated. And now these cell bodies would look pretty bright because I'm putting them sort of in the full dynamic range of my lookup table. And so this is something that we also see is if you play around with that display curve and you've sort of min-maxed it so that you're viewing gray values from 0 to 10, you think, oh my gosh, I have so much bleed through. I see these bright green cell bodies, but my probe shouldn't be labeling the cell bodies. Check that you're not sort of distorting your display curve, because ideally the real bright signal that's well labeled in your sample is filling the whole dynamic range and is not just existing as a few low gray value pixels down here. Now the alternative is that you could sort of fake it and pretend that you didn't have any of this bleed through. So you could shift that display curve over, and now it looks like, oh, I have no bleed through. I was using a really wide filter cube, a huge bandpass emission filter, but I didn't get any bleed through. And this might be okay if you're making a pretty cover image, or even if you report in your figure legend that this is what your display curve is. And it might be useful for thresholding an image to say that these are not values that you want to include in that threshold if you're doing some sort of segmentation analysis. But it's important to remember that showing a display curve like this or distorting your display curve like this so that you get rid of any bleed through that's actually there is sort of dishonest. And when I try and replicate your experiment, I'll think, wow, maybe I didn't choose the right conditions because I see a little bit more bleed through. So even if it's a small amount, it's okay to get rid of it if you're sort of forthcoming that you've adjusted your display curve in that way. And then let's take a look at our other example where we had maybe way more of the red probe, or maybe it had a much higher quantum yield than the green probe, and so we had a bit more bleed through to start with. And this might be what our range of gray values look like. So because we don't have as much green signal in the first place, we're not filling our dynamic range as much. And now what we see in our green channel from the real green signal is pretty close to what we see in the red channel. And so playing with these display curve tricks doesn't work as well. And you can imagine that doing some threshold-based segmentation would also not work super well because you're trying to divide between these very nearby gray values. And so filling your dynamic range is important. We sort of discussed this in the lecture 2 weeks ago. But especially when you're considering multiple colors, your ability to differentiate between those two colors. With some minimal amount of bleed through, you're better able to do that if you actually filter dynamic range. Okay, so let's go back to our original normalized curves at 1. We are collecting this green signal here, but remember, we still want to see the red cell bodies. We want a 2-channel image. And so let's imagine that we also collect window over the red emission curve. And you might do that alone. You might just repeat this whole process and just have a window over the red curve. And we could go through that step by step. But to not belabor the point, let's imagine a new scenario where we actually have a filter cube that has these 2 slots in it. So that emission filter has a little door to let this green light through and to let this red light through. And so that's pretty efficient because we can keep that same filter cube in place and we can collect both of those colors at the same time or in quick succession, just using 2 different excitation wavelengths. So this is pretty good. Seems great to save some time in our imaging, and we excite our green fluorescent protein, and this curve drops down, but now we have a new problem that this curve might have dropped down quite a lot, but now that we're collecting in this second door, in addition to the first one, we see a lot more of the red bleeding through into the green channel. But if we excite the red dye, then this curve goes up to its full height, where we're optimally exciting that dye. And the interesting thing is that even if we might excite some small amount of this green dye, this green fluorescent protein, we aren't going to get some backwards emission, right? So you can't excite here and get fluorescence here, because that would be a shorter wavelength of light, a higher energy, so you can't put in one energy and get more energy out, or if you do, you'll probably win a Nobel Prize. But this scenario is in some ways better than the one previously, because if you, let's say, have a problem where you have way more of the green fluorescent protein or green dye compared to the red dye, you don't see as much bleed through because you don't get that backwards excitation. Now you could still get some cross excitation of the green fluorescent protein, but it would have to have some emission that was higher than the wavelength that you excited it with. And so you end up getting less of the green to red bleed through in this scenario than what we saw previously. And so if you're stuck with a dye that has higher quantum yield, or maybe you can't get the secondary concentration down low enough, then you can play these tricks of sort of where do you put that really bright dye? Do you put it in the red or do you put it in the green? So even just changing, even just swapping your secondaries is a great quality control to check the amount of bleed through that you might have. Okay, and so just to give you some real examples of these kinds of excitation and emission filters. So here are 2 examples. One of a single bandpass, so focus on the red colors here, so those are the emission filters, so that's the dotted line boxes I was putting around those emission spectra. And so here, this is a single bandpass, so we're just collecting a band of wavelengths. This is a GFP filter cube, and this is a quad bandpass, so you're able to collect 4 of those little doors at once, and so you could just switch your excitation light 4 times, and you could keep that same filter cube in place. And this is great. It's what we have in one of our Axio scans, and so it's a very fast way to image 4 colors at once. But in the examples that I showed you, you can see how that might lead to more bleed through between channels. Another thing that I want you to notice is that we don't only see blue and red in these little bands. We also see all these weird spikes in the sort of infrared, far red part of our spectrum. And so it's important to look up, just like you look up the spectra of your dye or protein, you should also look up the specs of your filter cubes and see if you have some weird molecule that's fluorescing, that's emitting light at 900, and you start seeing this weird signal in your images, or maybe even you have some blue light excitation that you didn't expect. See if you have some holes in your filter that are red or UV, and that can sometimes explain some bleeding that you're getting between your channels. let's start by saying that, you know, we were exciting just at a single wavelength here, and that's how we sort of determined the amount of bleed through we were getting in our green and red channels. But in fact, that's not always the case. So I talked about an excitation filter, which was a band of wavelengths. And so if you say illuminate with some white light source, and then you put an excitation filter in place, you're not getting a single wavelength out, you're getting a band of wavelengths out. And so this line should actually be a bit wider. And you can see that now we're exciting sort of a band of the red dyes excitation as well. And so maybe this curve doesn't look like this, but it looks a little bit higher. We're exciting it a little bit more efficiently than we described before. And so even changing your light source from one that's a specific wavelength to a range of wavelengths can improve the amount of bleed through that you see. And there are examples of different light sources that do this better than others. So here's listed some lamp sources that you might have, some LED sources that you might have, or some laser illumination sources that you might have. And we have examples of these in the facility. And in the lecture 2 weeks from now, we'll talk in a bit more detail about different light sources, different types of illumination that you might use for different kinds of microscopy. And so we'll go a bit more in depth into not just for multicolor imaging, but the advantages and disadvantages to all these different light sources. But just for today, I want you to appreciate that the specificity, how narrow or wide these excitation sources are, can influence, even if you're using the filter, what kind of width of excitation you get and what kind of bleed through you get between your channels. Okay, so now let's improve our situation where we get rid of this second window. So if we really want to get rid of all bleed through, then we're not going to use a multi-band pass filter cube. We're going to use a single band pass. And we're going to use our narrow excitation. That's optimal here. But what if we narrowed our emission filter as well? So we narrowed our excitation, but we want to use some single wavelength of excitation. So couldn't we do the same for our emission? We could say we want to collect a single wavelength of emission. And the first problem with that is, of course, you would be collecting far less light. So you're going to have a dimmer image, you're going to have lower signal to noise. So the other problem is that what if you change the dye you're using tomorrow to a slightly different dye? Do you have to buy endless narrow band pass filter cubes for every single dye of interest? And this is something that you could do. It also would take longer to image because you have to move those filter cubes in another place each time. And it doesn't seem very efficient if you want to pack in 5 colors together. This example that I've been showing you has been 2 colors, but of course the desire is to image many overlapping spectra at once. And so we probably don't want to move in and out 8 different filter cubes in our microscope. And so one option is to use more spectral strategies. And in this scenario, this is a detection unit that we have in our 880, our LSM 880s. And in this case, what you can do, and I encourage you to go play with this, this little activity, where you can move these sliders and prisms to direct the light to either one of these PMTs or to this array of 34 channel PMT units. So if I move these in and out, you can see that it directs certain wavelengths of light, certain bands of my emission spectrum to different detectors in this scenario. And so this gives us a lot more flexibility to detect certain ranges of wavelengths and to move it around even within a given experiment. And we can move these in and out and be flexible, and we don't have to buy a new filter cube every time we want to image a different dye. There are other examples of this, a little bit more complicated geometries where you can direct light in different ways to different detectors. And then the last thing that I wanted you to remember about these kinds of examples is to not just look up the spectrum of your dyes that you're using, not just look up the specs of your filter cubes, but also of your detectors themselves. So you might have noticed on that diagram it described those PMTs as on that 34 array detector, which we'll go over in a second, as GASP PMTs. And you can see their efficiency at different wavelengths. And you can see that they're much more efficient than this other kind of PMT. But if you were imaging Alexa Fluor 750, that wouldn't be true. They have a much lower efficiency. It drops off dramatically in the near IR. And so it's important to consider what colors you're using, what dyes you're using, and even what detectors you then use to image those different colors. So where could you find all of this information? I'll go over near the end of the talk where you can find it specific for the HCBI's microscopes, but you might not only be using HCBI microscopes. So there's this great tool that's relatively new, created by Tally Lambert, who works over at the medical school. And he's created this great source for looking up the spectra of different dyes and proteins. And there are other spectra viewers out there. If you Google spectra viewer, you'll see all of your options. But I really like this one because it's fairly comprehensive, and he also allows you to shift the curves based on what your excitation source is, based on the quantum yield of your dye, and you can even build a whole microscope. You can add in filter cubes and excitation light sources, filters, and all of that. And so I encourage you, maybe it will be a project in the summer for us to build the HCBI microscopes in this FP base so that you could go in and see exactly how your dye would look in that microscope. But this is a great resource if you're trying to decide what is the overlap between these two dyes. Can I image them with a multi-band pass filter cube? Can I image them with this lamp excitation or this laser excitation? And what would the bleed through look like? And so I encourage you to go. There's nothing else to sort of see these firsthand. Okay, so let's look at some real examples. Maybe, is it okay if I turn the lights off, or is that bad?
Oh, we can do that.
Thank you. Okay, so some real image examples. So this is an example of a two-color image. We saw it in the beginning. We have some mitochondria labeled and some actin labeled. And you can see how much of the dynamic range we're filling down here. And you can see that I've adjusted these display curves. I've hit this min-max button so that we're only viewing from the min to the max of the gray values that a given channel encompassed. And so this looks pretty good. Like if I look at this image, you can clearly see where the mitochondria are and where the actin are. They look pretty well separated. And if I zoom in on the display curves here, what you can see is that in fact, like the example I showed you in cartoon form, there is actually a little spike here in the lowest part of this histogram, and that is from some of the red channel bleeding through into the green channel. And that's because I used a multi-band pass filter in order to image this. And so we have a little bit of bleed through, but maybe for your analysis, that qualitative view of the image is fine. The level of bleed through isn't a big deal. But what if I change the display curve like I showed you? So I'm sort of saturating this entire part of my histogram. Those gray values are all going to look the same. And I'm sort of boosting it up so that these seem more important than they are, more prevalent than they are. And if we look at our image, we see a lot of saturation. But you can also see that now it looks like we have a lot of mitochondrial signal right by the nucleus in the green channel that is truly there. But by boosting our display curve, we're sort of artificially making it look like the bleed through is worse than it is. And so you can imagine if maybe you didn't have your actin signal here and you tried to boost up your display curve and you hit min-max, and you say, oh no, I have all this bleed through. But what you don't realize is that it's such a small percentage of the rest of the gray values. It's such a lower gray value compared to what's really important. But what if you wanted to quantify this? You didn't want to just say qualitatively what the bleed through was. You could, for example, use a colocalization tool, which is present in the Zen software. Pretty easy just to click on it and get a color-coded image based on to show you colocalization. This is, of course, great if you are measuring colocalization, but if things are bleeding through into one another, they will also colocalize. And so these, the color coding here is based on some thresholds that are sort of hard to see. There's a horizontal threshold here, a vertical threshold here. And you can set these manually, but you can also do an automated method that does Pearson's correlation coefficient, calculates it iteratively on like the left and the right side of this threshold, or above and below this threshold. It tries to minimize the correlation to place that threshold appropriately. And once it's done that, these values here, it's calling your background, and then you have your what's calling here your vertical and horizontal thresholds here, and then anything above those thresholds is your co-localization. So in this case, we'll call it our bleed-through, and so anything that's pink is our bleed-through. So you can see that, like I said, we used a multi-band pass filter. We have a little bit of bleed-through in this image. And if you wanted to know, is that bleed-through, or maybe are these signals spatially co-localized, the best practice is to use single labeled samples. So you would have only actin labeled in another, only mitochondria labeled in another sample, and then you do the same analysis, set your thresholds the same, and see how much co-localization you see in those channels. And maybe you do have true co-localization, or maybe it's just bleed-through. But let's make this situation even worse. Let's imagine, oh sorry, I was going to zoom in on that to make it easier for you to see those values. So let's make the situation worse. So what I did is I added some of the red channel into the green channel. So I added some more bleed through, so you can see the histogram or the scatter plots were shifting over to the right, and you can see we have way more co-localization of bleed through here, where the mitochondrial signal is bleeding through into the green actin signal. And so what if this was your real scenario? How could you get rid of it? And of course, we've discussed how you might get rid of it by choosing different dyes, by making sure their concentration was appropriate, choosing ones with different, more similar brightnesses based on their quantum-Hiller efficiency. But what if you had some 10 fluorescent dyes, or maybe you had 2 that were chosen for you because you have some transgenic line that you can't get around? What if you really needed to fix this problem and you didn't have the option to change some sort of experimental protocol? Then you could do what we call spectral unmixing. And the examples I'm using here are 3 colors because it's easy to sort of see the colors and talk about bleed through. But I want to be clear that it's really rare that a 3-color experiment needs spectral unmixing. In many, many cases, you can fix any bleed through by choosing better dyes, changing the concentration of your probe, changing your imaging parameters, maybe going to that example where you could finally control what bands of emission light you were keeping, using specific excitations at the band of excitation. But in this scenario where you need to do spectral unmixing, this is what you would do. So the assumption of spectral unmixing is that you see some intensity in your image that's coming from many different fluorophores. So that would be your I value. So if you have 10 fluorophores, it's coming from an I of 10 different species in your sample. And you should have some reference spectrum for each of those fluorescent proteins or dyes. And that is going to be wavelength specific, of course. If you excite with different wavelengths, that reference spectrum is going to change. And that multiplied by the concentration of that given dye or protein summed up will give you the intensity that you see in your image, which is also wavelength dependent. Now this is true if you don't have any other non-linearities. So for example, you could have fret, you could have quenching, you could have saturation, and these will cause this to no longer be true. But if it is true, then all you have to do is measure this R spectrum for each of your species individually, and then you can calculate these concentrations, so the relative abundance of those different species based on the reference spectrum and the actual spectrum that you measure in your image. So ideally, you again have single labeled samples, and you take 5 or 10, however many species you have. The same number I of species you have is the number of reference spectra you should take in order to create these R values for this calculation. So single labeled samples, image them using the same conditions, and then image them all together, and you can then calculate these C terms here. So let's do this with the image that we had before, which is a pretty terrible idea because we have a 3-color image, and it's just a 3-channel image. We sort of hard to call it a spectrum because what you get out, if I measure this blue area or this red area or this green area, is a 3-point spectrum, which looks a lot different than the emission spectra that we saw before because we only have 3 values. So we're severely undersampling those emission spectra. And we're not using single labeled samples. So in this case, you can sort of get away with it because you have spatially separated, signals. So you have the nucleus, you have mitochondria, and you have these this actin signal that's spatially separated from your image. And so you can put your boxes over these different points and then run your spectral unmixing and get out a nice spectrally unmixed image. But this is not really ideal because you don't have a lot of data points here to do the spectral unmixing. And so it's really hard to tell on this projector, but you can get some strange artifacts when you're trying to assign pixels just based on these 3 values in your spectra. So what's a better way to do this? I want to compare. I want to compare 2 options that you might have done before in your imaging. So you can set up different numbers of tracks. So here I have a 3-channel image in both scenarios, but in one case I'm exciting all 3 of them at once, imaging them all at once. And in another case, I'm separating them into 3 separate tracks. So I'm turning on a given excitation light source and collecting this band of wavelengths. I'm turning on another light source, collecting a different band of wavelengths, and then again a third. And these are common methods to, it's similar to single band pass or multi-band pass filter cube, where you're trying to image things all together or separate them out temporarily. But there's a third option that's quite good for spectral unmixing. And this is really what you use if you have that 10-color image that you want to achieve. And that's, instead of using your tracks in this channel mode, you go into this lambda mode. And in this case, you can see that I'm not cutting this spectrum up into different chunks. I'm just saying collect all of it. And the way that I'm able to do that is using that 34-channel detection unit that we saw previously. So you're collecting this entire spectrum all at once, you're spreading it out and plopping it down onto 34 detectors in an array. And so in that way, you can get a 34-point spectrum instead of your 3-point spectrum that we saw previously. And when you do this, you get essentially a 34-channel image. So here, sorry, these wavelengths are sort of small, but we're going from blue to green to red here. And so you can see your nuclei pop up, and then you have your actin, and then you have your mitochondria at the end. And it's hard to see, but early on here you have nuclei bleeding through into your actin signals, and then later you have the mitochondria bleeding in, and then you finally get the mitochondria by themselves. And so you're exciting all 3 of these at once, and you're collecting this 34-channel image. So this is actually quite fast. You take a single image, a single pass of your laser, and get a 34-channel image. And then you can put them all together. If you assigned a color from blue to blue-green to green-red, all the way up into red, you would get an image like this. So this is not a 3-channel image, this is a 34-channel image, but we've assigned slightly different lookup tables to each of those 34 channels, and it gives you this image. And this is a much better image to perform spectral unmixing on, because if you select 3 points, so here we have our actin, there's a point here where we selected mitochondria, I think here is where we placed an X for the nucleus. You can see these spectra look more like spectra. And we have 34 data points in order to compose those spectra for our 3 channels, and then we can unmix it. And so this is really the ideal way to perform spectral unmixing if you have a 10-channel image. I'm sorry, I don't have a 10-channel image as an example here, but this is how you would perform that. And you'll notice there are little notches in these spectra, and that's because of the beam splitter that you use to send your laser light to your sample that reflects that wavelength that you're using, say your 488 laser, and it also blocks it from getting to your detector. So you'll have notches for each of your excitation wavelengths that are in your spectrum. So that's normally it would look like that. And so using this denser data, you can unmix your image into hopefully more than a 3-channel image. So how do you tell what microscopes have these capabilities, what microscopes might be better equipped to image certain dyes? So you can go onto our website under the Equipment tab, and each microscope is listed and has a brief description. And within that description, or frequently under that description, what we have listed are things like what filter sets do we have. So here in our Axioscan number 2, we have a quadruple bandpass filter, so you can image these 4 colors at once. And then we have a series of single bandpass filter cubes. And we even tell you, or Doug has told you, what the best dye combinations are. So it lists, if you're looking up, typically this is in reference to, say, like an Alexa dye that lists its excitation as peak excitation as its name. So Alexa 4488, for example. But anything that had a similar excitation spectrum, which you could find out on FP days, would be appropriate to use on this microscope. And then we have others like the LSM 880 confocal, which is described as having this 34-channel array detector, great for spectral unmixing. And it also tells you, in this case, what laser lines it has. So in this case, we're using that very specific excitation wavelength rather than a band that might be present in an excitation filter in something like the Axioscans. So depending on how many channels you have, depending on how much bleed-through is an issue, and if you have 1,000 slides versus 1 slide to image, that's how you can sort of make these decisions on what the optimal microscope is to use. So I think I'm at time, so I just wanted to leave with some final points. The first is look up the spectra of the dyes that you're interested in, put them all together on a plot, play with your excitation sources, play with your collection, what your filters are, and actually look at what the data might look like. In many cases, narrow excitation and narrow emission is great. Of course, you don't want to collect too little so that you get a dim image, but if you have a problem with bleed-through, narrowing what you collect or what you excite with is going to help you. You want to equalize your brightness, so don't be trying to image something that's 10,000 times brighter than something else. It's only going to make it harder. Use singly labeled samples. It's really rare that people bring in single labeled samples, but everyone is really worried about bleed-through, and that is the best way to see if you have signal bleeding into another. You can do all this co-localization analysis, but the end-all be-all is to label it with one color and see if that signal is truly in 2 structures or only in 1. And of course, there are other questions of antibodies labeling things that they shouldn't, which at the molecular biology level, we're across this, not molecular biologists. Actually, I am a molecular biologist. And then finally, and maybe most importantly, is keep your expectations reasonable and don't panic, because we saw how we could change those display curves and make it look like bleed-through was way worse than it actually was. And I think this is really important to remember that if you pull up any spectra on FP base, what you'll see is that there's overlap in almost all of them. And these are dyes that we frequently use in combination. And so you will be able to find a scenario or a change of the display curve that makes it look like you have bleed-through. But the question isn't whether there's any gray value of bleed-through, but it's really what are you quantifying in the end? And is the noise in your image, which could come from all the sources that we talked about a couple weeks ago, detector noise, but it can also come from any bleed-through. We can consider that noise if it's not the signal that we really want. What is that compared to your signal? And if your signal is high enough and quantifiable enough, then a small amount of bleed-through might be a compromise that you're able to make depending on what your quantification is down the line. And so with that, we'll finish and take any questions.
Thanks for the great talk, Erin. So what I don't understand is when we do linear unmixing, you say you also should bring the singly labeled sample, but when you get the equation, there is a summation of concentration into a standard reference intensity. So can you not just pull up the reference intensity from the reported dye and then do the unmixing without?
So that's exactly, I think if I understand correctly, that's exactly what I did here, where this was a single image,
Yeah.
and I clicked on points because these were spatially separated signals. So I could click on the actin or the nucleus or the mitochondria. But if you have very not spatially separated signals, then you wouldn't be able to do that.
There is an addition to that, is these reference spectra that you find in literature. They're acquired in ethanol and aerosol. They're not acquired inside your cells. And the spectrum, the emission spectrum of the fluorophores depends tremendously on things like Woodhouse potential, pH value, and so forth, that might be different inside your cells. The proportions between the little peaks you see in your cells are not the ones you find in the literature.
If you use linear mixing and selector region were not in the same, will it at all improve your signal noise?
So you can add, I didn't do it in this example, but you can add a background channel to this, and it will then create it as, in this case, fourth channel to your image. And so you could, subtract the background in that way. You are not able to do that if you have the 3-channel image and you try and add in a background channel. You can only unmix the number of channels you've collected. So if you've done this 34-channel collection and you really only have 3 colors or 10 colors even, then you can add in an 11th, which is your background. But you can't do that if you collected a traditional 3-channel image.
So why only 10 dyes? Because
I'm just saying that arbitrary number, and I actually don't know if anyone, I think people have unmixed 8. I don't know if we've done 10. You're welcome to try more. I mean, the idea is the number of data points that you have, these 34 data points, and the number of spectra you can fit in there, and what those dyes are that you're trying to fit next to each other. If you tried to put 10 dyes that were all centered at 500, you couldn't unmix 10 dyes. If you disperse them very nicely across those 34 channels, then maybe you'd have greater success. So that's a very arbitrary number. It would be very selection specific. So when you go to someplace like a spectra viewer, even if you're doing spectral unmixing, you'll see you are going to have spectra overlap. If you pile 10 into the visible spectrum, they're going to overlap, but you can still choose them. The other thing to consider is how you can excite, how many dyes you can excite. So in this scenario, you're exciting everything at once, right? And you need to have those lasers, you need to get that laser light to your sample. And so based on the hardware you have, you're limited in what channels you can excite, what dyes you can excite. And so you might instead choose, we were sort of going over this the other day, you might instead choose sort of pairs of 2 or even 3 in this part of the spectrum that are well excited by this wavelength, and then cluster them here and cluster them there, so they're not actually evenly dispersed, because you want to sort of match them with maybe 3 laser lines you're choosing to excite with.
maybe also the detector, right?
Yeah. So actually on the 880s, what we have is we have that 34-channel array, and then we have 2 more PMTs that will be more sensitive to that IR part of the spectrum. So you would typically add those in on either side, the blue and the red end of your spectrum, to collect sort of the edges. Okay. Well, I can answer questions after, but thank you so much.